everyone, welcome to a new session on molecular biology. Here we discuss spontaneous and induced mutations. Mutations can lead to changes in the structure of an encoded protein or complete loss in its expression. A change in the DNA sequence affects all copies of the encoded protein, hence mutations are damaging the cell or organism. Mutations can be both beneficial and detrimental. On the one hand, they are the source of all genetic variation, the raw material of evolution. On the other hand, most mutations have detrimental effects, and mutation is a source of many human diseases and disorders. Spontaneous mutations are the natural mutations occurring in low frequency without any definable cause. Induced mutations are the mutations artificially produced by the application of physical and chemical agents. The induction of mutations by artificial means is called mutagenesis. The chemical and physical agents which can induce mutations are called mutagens. Most mutagens are carcinogenic agents. Mutations for the most part are harmless except when they lead to cell death. Cells are continuously faced with endogenous stress and exogenous stress that can ultimately lead to DNA damage. To preserve genomic integrity, cells have an arsenal of repair proteins that detect different types of damage and initiate the appropriate repair pathway, or if irreparable, induce cell cycle arrest and or apoptosis. These responses need to be tightly regulated to ensure that repair pathways are only activated by genuine DNA damage and not, for example, in response to telomeres. This control is achieved by multiple levels of regulation, including checkpoint signaling, non-coding RNAs and post-translational modifications such as ubiquitination. Spontaneous mutation. Spontaneous mutations are those that occur without a known cause. They may be truly spontaneous, resulting from an inherent low level of metabolic errors, that is, mistakes during DNA replication, or they may actually be caused by mutagenic agents present in the environment. Mutations arise spontaneously at low frequency owing to the chemical instability of purine and pyrimidine bases and to errors during DNA replication. Spontaneous mutation rates in RNA virus whose genomes contain approximately 104 bases are roughly 1 per genome per replication for lytic viruses and roughly 0.1 per genome per replication for retroviruses and a retrotransposon. Mutation rates in higher eukaryotes are roughly 0.1 to 100 per genome per sexual generation. Spontaneous mutations on the molecular level can be caused by tautomerism, depurination, deamination and slipped strand mispairing due to replication errors. Spontaneous replication errors. Spontaneous DNA replication errors occur during mutation through wobble, in which non-complementary bases can pair because of the flexibility of the DNA helix. Mutations due to small insertions and deletions also may arise spontaneously in replication and crossing over. During normal crossing over, the homologous sequences of the two DNA molecules align, and crossing over produces no net change in the number of nucleotides in either molecule. Misaligned pairing may cause unequal crossing over, which results in one DNA molecule with an insertion and the other with a deletion. A common cause of spontaneous point mutations is the deamination of cytosine to uracil in the DNA double helix. Subsequent replication leads to a mutant daughter cell in which a TA base pair replaces the wild type CG base pair. Another cause of spontaneous mutations is copying errors during DNA replication. Although replication generally is carried out with high fidelity, errors occasionally occur. This figure illustrates how one type of copying error can produce a mutation. In the example shown, the mutant DNA contains 9 additional base pairs. Spontaneous chemical changes. Mutations can also result from spontaneous chemical changes in DNA. One such change is called depurination, the loss of a purine base from a nucleotide. This happens when the covalent bond connecting the purine to the carbon atom of the deoxyribose sugar breaks, producing an apurinic site. Another spontaneous chemical change that can occur is deamination, the loss of an amino group or NH2 from a base. This may occur spontaneously, but can also be induced by mutagenic chemicals. In this particular figure, you can see the loss of an amino group from cytosine produces uracil, as well as the loss of an amino group from 5-methyl cytosine produces thymine. This figure describes the mechanism by which uracil containing nucleotides are formed in DNA and removed in E. coli. Uracil is cleaved from the deoxyribose sugar by DNA uracil glycosylase. The deoxyribose with the uracil detached is then excised from the DNA backbone by another enzyme called AP endonuclease, and the gap is repaired. Tautomerization. All the 4 common bases of DNA have unusual tautomeric forms. The normal bases of DNA are usually present in the keto form. Mutation by tautomeric shifts in the bases of DNA are explained in the figure. In the example diagrammed, a guanine undergoes a tautomeric shift to its rare enol form G star at the time of replication. In its enol form, it pairs with thymine, and in the next replication, the guanine shifts back to its more stable keto form. The thymine incorporated opposite the enol form of guanine is also shown in the figure, where it directs the incorporation of adenine in the subsequent replication. The net result is a GC to AT mutation. If a guanine undergoes a tautomeric shift from the common keto form to the rare enol form at the time of incorporation as nucleoside triphosphate rather than in the template strand diagrammed, it will be incorporated opposite thymine in the template strand and cause an AAT to GC mutation. Tautomerization usually occurs by the transformation of the amino or NH2 group of adenine and cytosine to imino or NH group, amino-imino tautomerization, or by the transformation of the keto group of guanine and thymine to the enol group, which is also known as keto-enol tautomerization. The keto tautomer is called a lactam ring, whereas the enol tautomer is a lactim ring. The lactam or keto tautomer is the predominant form at neutral pH. The 4 bases of DNA can exist in at least 2 tautomeric forms as shown in the figure. Adenine and cytosine, which are cyclic amidines, can exist in either amino or imino forms, and guanine, thymine and uracil, which are cyclic amides, can exist in either lactam or lactim forms. The tautomeric forms of each base exist in equilibrium, but the amino and lactam tautomers are more stable and therefore predominant under the conditions found inside most cells. The rings remain unsaturated and planar in each tautomer. Human diseases caused by spontaneous mutations. Sickle cell anemia in humans is caused by a single missense mutation at codon 6 of the beta globin gene. As a result of this mutation, the glutamic acid at position 6 in the normal protein is changed to a valine in the mutant protein as shown in the figure. This alteration has a profound effect on hemoglobin, the oxygen carrier protein of erythrocytes, which consists of 2 alpha globin and 2 beta globin subunits. In this figure, you can see that sickle cell anemia is caused by missense mutation, wherein the codon GAG mutates to GTG and leads to a change from glutamine to valine. Retinoblastoma in children results from a germline mutation in one RB allele and a second somatically occurring mutation in the other RB allele. When an RB or retinoblastoma heterozygous retinal cell undergoes somatic mutation, it is left with no normal allele. As a result, the cell proliferates in an uncontrolled manner, giving rise to a retinal tumor. A second form of this disease, called sporadic retinoblastoma, results from two independent mutations disrupting both the retinoblastoma alleles. Since only one somatic mutation is required for tumor development in children with hereditary retinoblastoma, it occurs at a much higher frequency than the sporadic form, which requires acquisition of two independently occurring somatic mutations. Induced mutation. The artificial production of mutation is called mutagenesis, and the mutation inducing agents are called mutagens. Mutagens act at the molecular level, inflicting abnormal alterations on DNA. Mutagens are of two basic kinds, namely physical mutagens and chemical mutagens. In order to increase the frequency of mutation in experimental organisms, researchers often treat them with high doses of chemical mutagens or expose them to ionizing radiation. Mutations arising in response to such treatments are referred to as induced mutations. Generally, chemical mutagens induce point mutations, whereas ionizing radiation give rise to large chromosomal abnormalities. Physical mutagenesis. Physical mutagens mainly include the different kinds of radiations of the electromagnetic spectrum and also the products of the decay of the radioactive isotopes such as carbon 14, tritium or H3, S35, etc. The former includes short wave and high energy radiations such as UV rays, X-rays, gamma rays and cosmic rays. The latter includes alpha particles, beta particles and high energy protons and neutrons. Physical mutagens are two kinds, non-ionizing and ionizing. Non-ionizing radiation. Non-ionizing radiations include the UV rays, which range from 180 to 400 nanometer of the electromagnetic spectrum. UV rays generate a number of photoproducts in DNA. Two different lesions that occur at adjacent pyrimidine residues, the cyclobutane pyrimidine photodimer and the 6,4 photoproduct, have been most strongly correlated with mutagenesis. These lesions interfere with the normal base pairing, hence induction of the SOS system is required for mutagenesis. The insertion of the incorrect bases across from UV photoproducts is at the 3' position of the dimer and more frequently for 5' CC 3' and 5' TC 3' dimers. The cytosine to thymine transition is the most frequent mutation, but the other base substitutions called transversions and frame shifts also are stimulated by UV light, as are duplications and deletions. This figure describes the structure of a cyclobutane pyrimidine dimer. It also describes the structure of the 6,4 photoproduct. The structure forms most prevalently with the 5' CC 3' and 5' TC 3' between the C6 and the C4 positions of the two adjacent pyrimidines. The reversal of the mutagenic effect of UV radiation by visible light is called photoreactivation. The absence of inadequacy of the DNA repair mechanism in human beings leads to an autosomal recessive skin disease called Xeroderma pigmentosum. They develop skin cancer in those areas of the body that are uncovered and directly exposed to UV radiations. Ionizing radiations. Ionizing radiations include alpha, beta and gamma radiations, X-rays, protons, fast neutrons and slow neutrons. Of these, fast neutrons, thermal neutrons and X-rays are commonly applied for mutagenesis in plants. All of them are highly penetrating. They consist of highly energetic photons which can penetrate the cells and knock down the electrons of cellular atoms out of their orbit, leaving charged ions or free radicals. This results in the ionization of atoms. Here in this table, you can see some of the mutagenic ionizing radiations. Gamma rays, they are basically photons in nature and the source is radioactive elements. X-rays are also photons in nature and the source is X-ray machines. Beta rays exist as free electrons and the source is radioactive elements. Protons exist as hydrogen nuclei and the source is cyclotron. Alpha rays, the nature of alpha rays is helium nuclei and the source is radioactive elements cyclotrons. Neutrons exist as neutral particles and the source is cyclotron. Ionizing radiation results in the formation of ionized and excited molecules that can cause damage to cellular components and to DNA. Many different types of reactive oxygen species are produced, including superoxide radicals such as OH. The most biologically relevant reaction products are hydroxyl radical OH, superoxide radical O2- and hydrogen peroxide. These species can damage bases and cause different adducts and degradation products. Ionizing radiation can cause breakage of the N-glycosidic bond, leading to the formation of apyrimidinic or apurinic site, and can cause strand breaks that are responsible for most of the lethal effects of such radiation. Ionizing radiation can cause both gene mutations and chromosome breaks. Chromosome breaks may result in structural aberrations such as deletions, duplications, inversions and translocations. In general, the rate of gene mutation is directly proportional to the radiation dose. This linear relation between radiation dose and mutagenesis indicates that a single radiation hit is enough to produce a gene mutation. The efficiency of effectiveness of radiations in mutagenesis is related to several factors such as stage of cell division, cellular level of oxygen, temperature, moisture content of the cell, etc. Cells in the diplonema and dikinesis stage of meiosis I are more prone to radiation induced mutations. Higher concentration of oxygen and reduced water content are also ideal for mutagenesis by irradiation. Chemical mutagenesis. A good number of chemicals are reported to be mutagenic. Many agricultural chemicals such as insecticides, fungicides and herbicides are found to induce gene mutations as well as chromosomal aberrations. The important chemical mutagens include base analogues, alkylating agents, acridine dyes, deaminating agents, hydroxylating agents, etc. Of these, base analogues act only on replicating DNA and the others act on both replicating and non-replicating DNAs. Base analogues. Base analogues are compounds very similar to nitrogenous bases. They are the derivatives of normal bases, so they can be used to replace the normal bases in the DNA for producing transitional substitution mutations. Artificially synthesized base analogues are 5-bromouracil, 5-chlorouracil, 5-iodouracil, 2-aminopurine, 2,6-diaminopurine, etc. Of these, 5-bromouracil and 2-aminopurine are the most commonly used ones. For example, 5-bromouracil is an analog of thymine that has bromine at the C5 position in place of the CH3 group found in thymine. This change does not affect the atoms that take part in hydrogen bonding in base pairing, but the presence of the bromine significantly alters the distribution of electrons in the base. The normal structure or the keto form of 5-bromouracil pairs with adenine as shown in the figure. 5-bromouracil can frequently change to either the enol form or an ionized form. The latter pairs in vivo with guanine. Thus, the nature of the pair formed in replication will depend on the form of 5-bromo-uracil at the moment of pairing. Here in this figure you can see 5-bromo-uracil causes transitions almost exclusively. This figure describes the mechanism of 5-bromo-uracil mutagenesis. 5-bromo-uracil causes mutations when it is incorporated in one form and then shifts to another form. In its normal keto state, 5-bromo-uracil pairs like thymine. Thus, 5-bromo-uracil is incorporated across from adenine and subsequently mispairs with guanine, resulting in AT to GC transitions. In its ionized form, 5-bromo-uracil pairs like cytosine to form 5-bromo-uracil C. Thus, 5-BU is misincorporated across from guanine and subsequently pairs with adenine, resulting in GC to AT transitions. Another analog widely used in research is 2-aminopurine, which is an analog of adenine that can pair with thymine, but can also mispair with cytosine when protonated, as shown in the figure. Therefore, when 2-aminopurine is incorporated into DNA by pairing with thymine, it can generate AT to GC transitions by mispairing with cytosine in subsequent replications. Or if 2-aminopurine is incorporated by mispairing with cytosine, then GC to AT transitions will result when it pairs with thymine. Genetic studies have shown that 2-aminopurine like 5-bromo-uracil is very specific for transitions. This figure describes the alternative pairing possibilities for 2-aminopurine, an analog of adenine. Normally, 2-aminopurine pairs with thymine, but in its protonated state it can pair with cytosine. Alkylating agents. Alkylating agents are the chemicals which can transfer their highly reactive alkyl groups such as methyl or ethyl groups to the phosphate groups or bases of DNA. Ethyl ethane sulfonate or EES, ethyl methane sulfonate or EMS, methyl methane sulfonate or MMS, dimethyl sulfonate or DMS, etc. are alkylating agents. Alkylation of the phosphate residues and bases produces substitution mutation, frame shift mutations and chromosomal aberrations in the following ways. Alkylation of the phosphate residues interferes with DNA replication and inflicts breaks on the sugar phosphate backbone. This may produce phosphate triesters and induce gene mutation. The alkylation product of guanine or 7-ethyl guanine often pairs with thymine, that is replacing adenine, producing a transitional substitution mutation. During the repair of alkylated bases, errors sometimes occur. This produces transitional and transversional substitution mutations or even frame shift mutations. Some alkylating agents bring about the cross-linking of DNA strands or even DNA molecules. This in turn prevents DNA replication, induces chromosomal aberrations and produces substitution mutation and frame shift mutations. For example, ethyl methane sulfonate, a commonly used mutagen, alkylates guanine in DNA forming O6-ethyl guanine. During subsequent DNA replication, O6-ethyl guanine directs incorporation of deoxythymidylate, not deoxycytidylate, resulting in the formation of mutant cells in which a GC base pair is replaced with an AT base pair. In this figure, you can see the EMS alkylates guanine at the oxygen on position 6 of the purine ring forming O6-ethyl guanine, which base pairs with thymine. 2 rounds of DNA replication of a strand containing ETG yields a mutant DNA in which a GC base pair is replaced with an AT pair. Deaminating agents. Nitrous oxide reacts with bases containing amino groups. It can change the structure of such bases by deamination. It can bring about the oxidative deamination of adenine guanine and cytosine producing hypoxanthine and uracil respectively result in transitional substitutions. On the other hand, xanthine pairs with cytosine producing a machine pair. Intercalating agents producing distortions in DNA. The intercalating agents form another important class of DNA modifiers. This group of compounds includes proflavin, acridine orange and a class of chemicals termed ICR compounds. These agents are planar molecules which mimic base pairs and are able to slip themselves or intercalate between the stacked nitrogen bases at the core of the DNA double helix. In this intercalated position, the agent can cause single nucleotide pair insertions or deletions. This figure shows the structures of the common intercalating agents such as proflavin, acridine orange and ICR 191. An intercalating agent slips between the nitrogenous bases stacked at the center of the DNA molecule. Hydroxylating agents. Hydroxylamine NH2OH is very specific in its action. It reacts mainly with cytosine and guanine and brings about transitions and mispairing. This produces hydroxylaminocytosine which pairs not with guanine but with adenine. Thus, hydroxylamine induces a transitional substitution. Other chemical mutagens. They include nitrosoguanidide, formaldehyde, mustard gas or dichlorodiethyl sulfide, hydrazine, hydrogen peroxide, etc. Let us now summarize what we have learnt in this session. The development and function of an organism is in large part controlled by genes. Mutations can lead to changes in the structure of an encoded protein or to a decrease or complete loss in its expression. Mutations are alterations in DNA sequences that result in changes in the structure of a gene. A mutation involving a change in a single base pair, often called a point mutation. Treatment with ionizing radiation or various chemical agents increases the frequency of mutations. Mutations arise spontaneously at low frequency owing to the chemical instability of purine and pyrimidine bases and to errors during DNA replication. A common cause of the spontaneous point mutations is the deamination of cytosine to uracil in the DNA double helix. Subsequent replication leads to a mutant daughter cell in which a TA base pair replaces the wild type CG base pair. Another cause of spontaneous mutations is copying errors during DNA replication. Although replication generally is carried out with high fidelity, errors usually occur. Natural exposure of an organism to certain environmental factors such as ultraviolet light and chemical carcinogens can cause mutations. Mutation produced artificially by non-biological agents are called induced mutations. The artificial production of mutation is called mutagenesis and the mutation inducing agents are called mutagens. Mutagens act at the molecular level, inflicting abnormal alterations on DNA. Mutagens are of two basic kinds, namely physical mutagens and chemical mutagens. Generally, chemical mutagens induce point mutations, whereas ionizing radiations give rise to large chromosomal abnormalities. Here are a few questions for you to work out. 1. Discuss the SOS induction system which allows the cell to trade death for a certain level of mutagenesis. 2. Discuss the role of aflatoxin B1 in mutation. 3. How the hydroxylamine causes mutation. 4. Differentiate between ionizing and non-ionizing radiations. 5. Explain how mutation helps in evolution. Here I can suggest a few books for your reference. Essentials of Cell Biology, an Introduction to the Molecular Biology of Cells, written by Alberts B. Bray D, Johnson A, Lewis J, Raff M, Roberts K and Walter P. International Student Edition, Garland Publishing Incorporated, USA, 1998. Genetics, a Conceptual Approach, written by Pierce B. A., W. H. Freeman and Company, 2007. Evolutionary Genetics, written by Smith J. M., Butler and Tanner Limited, 1998. Genes 7, written by Lewin B., Oxford University Press, Oxford, 2000. Molecular Cell Biology 4th edition, written by Harvey Lodish, Arnold Burke, S. Lawrence Sipersky, Paul Matsudaira, David Baltimore and James Darnell, published by W. H. Freeman and Company, New York, 2000. Modern Genetic Analysis, written by Griffiths A. J. F., Gelbert W. M., Miller J. H., published by W. H. Freeman and Company, New York, 1999. Here are a few websites for your reference: www.biology.lifec.org, www.brighthub.com/science/genetics/articles, www.ncbi.nlm.nih.gov/books NBK 21475. Thank you for watching this program. Let's meet in the next session with a new topic. Until then, bye.